Welcome, everyone, to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Increasing Throughput in Protein Thermal Shift Assays. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Protein stability screening is often an integral component of the drug discovery process, enabling researchers to determine binding affinity and efficiency in the early stages of the compound screening process, thus allowing investigators to advance only the most promising proteins through to the next phases of development. Historically, protein interaction and stability have been measured using a variety of techniques, such as NMR, differential scanning calorimetry, and surface plasma resonance imaging. Typically, these methods lack the scalability for the fast-paced drug discovery sector and often require large amounts of precious sample. Over the past several years, thermal shift assays have developed exponentially in order to provide a high sensitivity with the speed and versatility that the biopharmaceutical industry has been demanding. Our speaker today, Rama Badugu, is an application support consultant at Roche Life Sciences and Roche Diagnostics. Dr. Badugu will highlight the specifics of the thermal shift assay and describe strategies for employing it as a high-throughput screening method while utilizing Roche's LightCycler 1536 real-time PCR system. Before our speakers get started, I want to encourage you to submit questions for our Q&A session at the end of the presentation. We will try to answer as many questions as we can. Simply type your questions into the Q&A box on the left-hand side of your screen. Additionally, I'd like to ask everyone now to disable their pop-up blockers, since we're going to have 3 short polls that will deploy during the presentation. Don't worry, though, I'll cue the audience just before the deployment of each poll. So, without any further ado, let's get our webinar underway. Rama, you have the floor.
Thank you, Jeff. I would like to thank Jen as well for hosting this webinar. It is a great opportunity for Roche to actually present the thermal shift technology on the LightCycler platform. So without wasting your time, I'll go ahead and start the process. The idea of thermal shift assays on LightCycler instrumentation. Before we go ahead and talk about the details, I just want to give an important notice here stating that the instrumentation that we provide is basically used in the research use only and not used in any diagnostic procedures. So what are thermal shift assays and how exactly do one use in their workflow? So thermal shift assays are the latest assays that could be used in understanding the interaction between a protein and a protein and a protein and a ligand as well. The whole thing started off somewhere around 1990, and people have been using it with NMR. ITC is one approach, and CD spectra is another approach. But there were some issues with the technologies that were used previously, and mostly about the throughput, in the sense like what you see in most of the biological applications in these days. We are planning to actually maximize the ability to detect these interactions and screen those molecules for your downstream drug screening approaches. And that exactly is what happening even in the thermal shift assays as well. As you see in this slide, as we progress from 1990 to 2015, what we basically did was we basically reduced the time to get to the answer, and also reduce the protein that is required to do these kind of assays. And but in a very simplistic form, I just want to go through exactly what these thermal shift assays are. Like what you're seeing here is basically a pictorial depiction of what exactly happens in the well. These are the fluorescence-based approaches to detect thermal shift, also known as TSA in other words. This whole phenomena is basically built on 2 activities. One would be if a ligand interacts with a protein, it contributes to the thermodynamic stability of the protein. So that is pretty important. And the second thing would be ability of recording this event by using a reporter dye. What the most popular one is the super orange. In the first part of the picture, what you're seeing is that you would be finding a native protein, and as we provide heat, it unfolds. And then this unfolding exposes the hydrophobic natures of the protein. And these hydrophobic pockets would allow the super orange to bind to those hydrophobic pockets. And super orange being a non-discriminative association with these hydrophobic pockets, we basically would be recording this as a fluorescence event on a detector. And the super orange by nature, when it is present in an aqueous solution, the fluorescence is low, but when it is associated with the hydrophobic pocket, then there is a tremendous increase in the fluorescence. And this is basically recorded by the detector. And to do this process of actually picking up the fluorescence information, you need really good instrumentation to do that activity.
Okay, so the first of our poll questions has come out, and it's asking the audience, are you currently performing thermal shift assays? And we're going to have a follow-up to that question, so that one's pretty straightforward and quick. So the next of our poll questions is going to be, if you answered yes to the previous question, at what throughput? Are you using them at 96 well, 384 well, 1536 well, or greater? So we'll give a couple seconds for everyone to answer. And Rama, you can continue.
So as I was saying, the importance of the instrumentation is basically the ability to actually provide good heat source for the reaction and the ability to detect that event by a detector without any time delay. LightCycler instrumentation has got two important components. One would be the ability of detecting the fluorescence event and the heat source. And we have introduced the LightCycler 480 system way back in 2005. It's almost a decade in the market. And the hallmark of the LightCycler instrumentation started off somewhere in 1998. We were able to do this recording of the fluorescence events in the PCR scale as well. It's very rapid and reproducible PCR conditions. Because of the way that we design the instrumentation and the associated QC process, we can guarantee well-to-well, plate-to-plate, and instrument-to-instrument consistency in all the portfolio of the LightCycler instruments. And the two important components that we actually see in this instrumentation is basically the heat source as well as the detector. These, I call them the heart and the soul of the instrument. First, onto the right-hand side, you would be seeing a block picture. And this is basically the heat source that provides the heat to the reaction. We have a patented technology, what we call it as a thermobase. The objective of the thermobase is basically to distribute the heat all across the plate evenly. In other words, like if we have one reaction and split it up into two components and put one reaction or one component in well A1 and the other component in P24 in a 384 format, if you run the reactions, then they are so aligned together in a really tight fashion. The standard deviation between them should be something like 0.01 in terms of the alignment. And this feature is also provided with the help of the detector. We are not a scanning instrument. We don't go from well A1 to A2 to A3. We take all 96 wells at the same time, all 384, in fact, all 1,536 reactions at the same time. So we do not waste time in collecting the fluorescence events. And that becomes a factor when you're actually doing your assays, because the time delays, you might be missing some of the information that would not be captured at the right time.
Okay, our third poll question is coming up at this time, and we're asking, at this point in time, is increasing throughput for your TSA assays important to you? So I'll give you a few seconds to answer that one. All right, Rama, I think we can continue.
As I said in the previous slide, the ability of the instrument is basically to provide a reliable heat source and also collect the fluorescence events without any time delays. We devised an assay in which we wanted to study the uniformity of TM all across the plate. And when I use this word TM, what it tells you is basically the amount of the protein that has basically unfolded and has been accessible to the supraorange and that has been detected by our detector. And this uniformity assay, we did it on a 96-well plate. And what you're seeing here is a pictorial representation of all the TMs in the 96 well. And as you can see, the standard deviation is close to 0.2, which is actually talking about the resolution of the instrument in the sense like it can actually pick up a deviation as low as 2, which would be actually in the ability of detecting a resolution between your compound A versus compound B. And with this approach, what we did was we wanted to move it a step further, not only looking at the TMs on the protein with the supraorange, but we wanted to see what would be the delta TM, which is actually the presence with the compound. So we do have one column dedicated for protein plus supraorange, and the other columns are basically dedicated with the differential concentrations of the protein. We used the conventionally expressed large-scale expression of the protein, and we used around the 5 micromolar with the 20x concentration of the supraorange, the reporter molecule. And the ligand, we started off with the 4 millimolar concentration all the way to 250 nanomolar concentration with a 2-time dilution series. And we do have a 0 micromolar concentration as well to depict the reference column. We did this almost like 24 replicates on the plate, and this is on a 384 system. And in the data, you might be seeing a couple of outliers, and usually you would be having something like 0 to 4 outliers, and it depends upon your pipetting ability and things like that. Usually, if the reaction is present, then it would actually show up the data for you. Now, let us look at the data. What you're seeing here is the top one is a pictorial representation of the fluorescence curves as recorded during the reaction. And mind you, these reactions are basically around not more than 10 minutes in the sense like you can actually screen all your 384 wells within 10 minutes. And but the lower portion of the data representation as an Excel sheet with the conditional formatting, you might be seeing that it is pretty difficult to see the figures. So what I did was I went ahead and put it as a separate slide. And in this slide, the numbers are pretty clear. I want you to concentrate on the fact that the standard deviations in all these replicates are pretty tight and close to 0.01. And what this says is that the consistency of the temperature and the fluorescence that is recorded across the column. And you have a zero concentration of your ligand on the last column on the right-hand side, and the highest concentration is seen on the left-hand side. And you would be seeing that there is a temperature difference of almost 18 degrees with a resolution of 0.2 degrees. These days, most of the biological assays are actually trying to figure out that subtle differences. And this instrument is actually capable of picking up that kind of a difference where it is only a difference of something like 0.1 or 0.2. And with the help of the 480 data, what we did was we wanted to extend this and see if we can actually record these kind of events on a 1536 instrument. Remember, the 1536 instrument has got 1,536 reactions to be run within the 10 minutes time span. And the volume of these reactions are also pretty low. So it was a very interesting experiment to do. Before going about showing about the data on the 1536, I wanted to give you an introduction to the 1536 instrument as such. It is the only plate-based qPCR system with a 1536 well format. There is no other instrument that can actually match with the throughput that the 1536 can provide. And it is actually established with the same architecture as we designed the LightCycler 480. For example, the 480 data that you have seen in the protein melt in the previous slide, we wanted to match those things on the 1536 as well. Lower cost per reaction, you are basically reducing the reaction volume. The maximum volume that you would be putting in the 1536 well is around 2 microliters, which actually helps you in two fashions. One would be a high throughput, which actually increases the number or maximizes the number of the molecules that you want to screen, and also actually reduce the cost of the reaction. One would expect that a format of this nature would be having an accessibility for automation for a complete walkaway workflow. And the 1536 actually comes with an automation protocol or software on it. So, and we have an experience with working with the different automation companies in integrating the 1536 instrument with their automation protocol, and then actually controlling the 1536 instrument with the scheduling software provided by the automation group. So how does the data look? So before going into the data, I would want to share you how the assessment of the instrument is done. So with our experience in the applications lab, we have tested a number of instruments and came out with a very good understanding that most of the instruments are pretty good at actually looking at the temperature ranges between something like 40 and 75. Most of the instrument can do that kind of an activity. But we wanted to see how does the 1536 instrument actually performs in the lower range, like around 50s, and also around higher ranges, like around 75s, where most of the instruments fall apart, in the sense like because of the lack of ability to hold the temperatures properly or lack of ability of collecting the fluorescence data at the right time. So we have designed an experiment in such a way that we did have some data generated by experiments that were created by us. We would be actually looking at the protein melt behavior somewhere around 75, and then also around 40 and between 40 and 50. And we wanted to do a correlation plot and see how 480, which we know does a really good job with the protein melt, would actually reflect in the 1536 data as well. So what you are seeing in the first correlation plot here is the linearity in the TM and around 8 degrees between 1536 and the 480, with an R-square value of close to 0.99. And this data is a representation of 16 individual experiments. With the temperature ranges between 50 and 58, we were able to see a really good correlation with 480 and the 1536, which is basically telling us that the architecture that was used in building a 480 was being faithfully replicated on a 1536 format as well, in spite of actually reducing the volume of the reaction and also reducing the cost of the reaction. So this was very good at the lower temperatures. Now we want to move it into the higher temperature ranges and see. So we did the similar experiments with other data set. And again, we were able to see a linearity over 5 degrees between 70 and 75. Again, an R-square value of close to 0.99. And again, it's an experiment 16 times have been replicated to get this kind of an information. So this also suggests the same thing, that it is even at the lower ranges and the higher ranges, we have a consistency in terms of representation of what we have seen on a 480 scale as well as a 1536 scale. So what are the advantages of moving your screens onto this kind of a phenomenon like on the 1536 scale? It's quick. One thing is it's very quick. the technology actually, when you compare from NMR to all the way to thermal shift assays based on fluorescence, the time to get to the results have been reduced like probably 1,000 folds. And what it actually helps in terms of a user is basically a user would not have to worry about the protein stability that they are actually planning to screen for different compounds. Like even if it is a really unstable protein, because of the quick nature of getting to the data, we can actually use relatively less unstable proteins as well for screening. It is the same performance in terms of the data that you get from different other techniques like SPRI or, for example, in CD spectra. Most often what happens is that whenever we bring in a new technology into the market, we need to either match the existing technology or we need to be better than the existing technology. And I think the TSA or the thermal shift assays on a qPCR instrument like LightCycler product line actually brings in the same, or in some instance even better information, where you're actually looking at close to 0.2 degrees in difference in terms of resolution, is basically a step forward in the right direction. High throughput options, and what this does is basically it will give you a ligand screening project that you can do it on a 1536 scale. I mean, that kind of ability is provided only by 1536 instrument, because we are the only 1536 instrument in the market for a plate-based system. You will also have a continuity, not necessarily that you are actually using all 1536 wells. Let us say that you have started off with a 96 system, and then as you are progressing in terms of your compound library, you can actually move from 96 to 384 and to 1536. And using the same technology, like you would be starting off at a 480 scale and then moving into the 1536 scale to actually look at your compound screen. Very low volume reactions require micrograms of protein. You really don't have to do huge, large-scale protein isolation projects to do your thermal shift assays. Because of the size of the volume in terms of the reaction, it would be basically easy to get away with really low amounts of protein and get your data quickly. Multiple variables can be introduced into the same reaction. This is one feature that would not be available in lower throughput, let's say 96 format. Because of the real estate that you have on a 1536 plate, you can actually create an experiment that would have multiple variables on the same plate. For example, most of the decision points that the researcher has to make is like, should I go for maximization of the protein or should I go for the maximization of the ligand or the compound that they are screening? In fact, because you have a 1536 format available, you have a better real estate to actually do your experiment. You can actually create an experiment where you can accommodate both the maximization of the protein as well as the maximization of your ligands as well. Higher resolution in data point, that is the single most objective of the instrument, is that we are the only one in the market that can capture more pictures of your 1536 plate in 1 degree difference. What it really means is that if there is a subtle change, and that is the reason why you can actually pick up that the standard deviation of 0.2 degrees, is because of this ability of the LightCycler portfolio to actually capture those subtle changes. We can pack in more number of data points within that 1 degree change. So these are the advantages that you would be having when you are actually doing a thermal shift assay on a LightCycler 1536 scale. In total summary, I want you to take away 3 important points that are associated with this instrumentation. It's the ability to capture more data points per degree change, which means to more resolution to the users, which means that you can actually see really subtle changes and probably identify better molecules in your screen. And the second point is exceptional control on temperature ramp, a linearity over 8 degrees in lower temperature, and a consistency at the higher temperature. What it tells you is that we designed an instrument that is basically capable of holding that lower temperatures properly and also ramp up in the higher temperatures much better than rest of the instruments in the market. What this helps in terms of a researcher use is basically studies full spectrum of your compound library, and that too fast within 10 minutes. Full spectrum of compound libraries, I have seen scenarios where researchers have used systems where the ability of holding the temperature at the lower temperature range is pretty difficult, and they have lost some of the valuable molecules in the screen just because of that inability, which we are going to fill that gap. And in the 1536 showing the same characteristics as the 480, what you can do with this approach is basically you will have the continuity of your data from 480 to the 1536 level. And not only that, but you are having a higher throughput at a lower cost per reaction. That's the single most advantageous where you can actually screen your compounds for less, and also, as I said previously, pretty fast. All this work is basically done with a really good collaboration that we developed with Chuga in Japan, Broad University, Duke University, and the University of Texas. These things are not possible without this collaboration. I would want to extend my thank you to all my collaborators, and thanks for listening to my talk.
Great. Thanks, Rama. That was a great presentation. I think you gave us all some really great insight into the versatility of the thermal shift assay, and I think you made a great case for the LightCycler 1536 being an essential tool for anyone doing protein drug discovery work. So we thank you for that. All right, everyone. Before we start the Q&A session, I want to let everyone know this is your final chance to submit questions for Rama, so hurry up and submit them now. And with that, let's begin our Q&A session. I'm starting to see that there's a few questions beginning to stack up, so why don't we start answering a few of them, and we'll try to get through as many as we can.
Okay, so just before we get to our first question, I'd like to go over the polling results that we had during the talk. And so for the first poll question, we had asked everyone, are you currently performing thermal shift assays? And 66% of you said you weren't. 33% said you were. Of those that were doing thermal shift assays, about 50% of you said that you were using 384 well format, where almost 40% of you said the 96 well format, and then the rest evenly split between 1536 or greater. And then the final question is whether or not you were interested in increasing throughput for your assays and whether that was important to you. And almost 52% of you said they were. So I think with that, Rama, I don't know if you had any comments on that, or we can just get right to the first question.
Yeah, I'm encouraged actually looking at the poll questions as the member of audience were interested in actually increasing the throughput of the assays here, and that's pretty encouraging for us. Yeah, let's go with the questions.
Great. So the first question is, you mentioned during your presentation that the LightCycler 1536 is automation friendly. Can you elaborate on this? And can you share any examples of where this has been successfully implemented with automation?
Yeah, right around 2009, when we looked at when it took a serious look at the 1536 instrumentation, we came up with an API module. So what it does is that it is basically helps the users to integrate their stacking as well as liquid handler, or in combination of both, to the LightCycler 1536 instrument. Advantage of that is basically the plate slacker, the scheduling software can actually control the LightCycler 1536 instrument. And utility value of this activity is basically to do a complete walk-away automation, in the sense the user can actually program and then put the plates inside the stacker, and once it goes in, then everything would be done by the liquid handler or the stacker, or a combination of both of them. And the one example that is pretty obvious right now, and that we know that has worked very well, is in Boehringer Ingelheim in Connecticut, where they have the thermo orbiter with the Momenta software integrated to the LightCycler 1536, and it is basically doing an awesome job. Not only that, but we do have a lot of experience in using different automation-friendly softwares. We know that it works in Access. We know it works in even in different situations where you are actually starting off with a liquid handler as opposed to just a stacker.
Okay, great. So let's move on to the next question. It said someone asked, you mentioned resolution of the data in your presentation. What does this mean?
That's a very, very interesting question, and it's also an important question. And the reason why I say is that the ability of capturing the fluorescence events over a very short temperature range, and that is possible only in the LightCycler product portfolio. That's basically because the way we have designed the block and the way we communicate that information to the detector. And unlike the other instruments where you actually, so the ramping for other instrument means that you need to go to a certain temperature, stay in that temperature before the block gets equilibrated, then go to the next stage, and then stay in that temperature before the block gets equilibrated. But in the LightCycler instrumentation, the equilibration of the temperature on the block is an instantaneous feature, just because of the thermal base that we have that distributes the heat evenly all across the plate. And that actually reduces the time in melting curves, and that actually helps in getting that information, the protein melting information, in a very short period of time. The runs typically would be something like close to 10 to 15 minutes. And imagine doing 1536 data points in 15 minutes, and the number of data points you can acquire out of that things. So it's a very fine-tuned system where you can actually capture many number of fluorescence events in a very short temperature range.
All right. So on to our next question. One of our audience members would like to know, can the LightCycler 1536 do anything other than thermal shift assays?
Absolutely. So the thermal shift assays as an application came as a second component to the instrument. And initially, the LightCycler product portfolio itself is basically for the quantitative qPCR, and it is built for the amplification of DNA and then reporting by probably cyber-green or hydrolysis probes and hybridization probes for that matter, and capturing that information and providing that as an amplification curve to the user. And later on, the thermal shift assays have been added into the as an application into the portfolio, and it can be done in all the instruments that we have in the LightCycler portfolio. And 1536 is the major one, giving you the high throughput as well as high visibility into the data points by higher resolution.
All right. So one of our audience members also wants to know, I guess it was about one of your slides, what about wells A2 and E20? Why were they blank, and were the blanks included in the standard deviation?
Yeah, that's a good point. Whoever saw that, I mean, they're pretty detail-oriented person for sure. Actually, A2 and E20 were missed because of the pipetting mechanism. I think that was overblown, and that's the reason we took it out of the calculations. And the A2 and E20 are not part of the standard deviation at all. Yeah.
All right. Someone else would like to know, what is the typical concentration of antibody you would use for a thermal shift formulation screen?
It's an interesting question. Specifically, we in the applications lab at Roche, we do not have a hands-on experience with an antibody screen, but usually it depends upon the, for example, the protein and the ligand interactions. It depends a lot on the number of the hydrophobic patches that are available on the protein, because that gives us a better fluorescence event that need to be captured, and that can actually give us a better understanding what would be the delta Tm between after interaction and before interaction situation. But it needs to be empirically determined, and in an antibody screen, unfortunately, we don't have a hands-on experience. But that can be determined pretty easily when you basically do a matrix of the super concentration versus your antibody concentration, and it can be easily determined, and it would not take more than 10 minutes of your time to get that thing done.
All right. So one of our audience members also would like to know, they had said, it sounded like you said 2 microliters is the maximum volume in the 1536 format. Don't you mean minimum volume? What volume was used for the 1536 data?
The 1536 data that we showed on the slides, that would be the 2 microliter volume, and 2 microliters is the max volume, not the minimum volume. And minimum volume, I mean, you can go down to 1 microliter and 1.5 microliter. The detector is capable of seeing those fluorescence events as well. But again, you are actually playing at the level of ability of providing fluorescence information to the detector. And most of our standardized experimentation, especially for qPCR, as well as for the protein melt assays that we have performed, we have done it at 2 microliters.
All right. Someone asks, what are you using for sample dispensing? Also, what was your ramp rate for the temperature gradient?
Ramp rate for the temperature gradient. I mean, this is a collation of multiple experiments. I'm not sure exactly onto which experiment the user is actually pointing to, but usually the default ramp rates are more than enough for for these experiments. LightCycler 480 and even the LightCycler 1536, right around 4.8 and 4.4 is the temperature at which it ramps, and we are the fastest rampers in the market. There is no instrument, at least on surface of Earth, that can actually ramp at that level. And what was the second point, Jeff?
Give me 1 second here to find the question again. They asked, what are you using for sample dispensing, as well as what is the ramp rate?
Yeah, sample dispensing. There are multiple liquid handling companies we actually interact with, and on the field, I have seen a number of users using different kinds of dispensing instrument. For example, if you're doing a lot of bulk dispensing, especially when you had your ligand already stamped by, let's say SciBio with a Vario 1536 head, you can actually stamp your ligand plate directly onto a 1536 plate. It would not take more than 2 seconds. And then use your bulk dispenser, for example, Cobra, with a 4-channel and dispense your protein of interest. And that can be done probably within not more than 6 or 4 minutes. There are other instruments as well. For example, LabSite, the non-touch liquid dispensing, even that is possible. Again, the speeds vary depending upon what kind of destination and the source plate you are using in terms of whether it's a 1536 to 1536 transfer, whether it is a 384 to 1536 transfer. But in our situation, the one that we have used is basically Cobra and then the 1536 plate.
Okay. Someone would like to know, how many data points are you recording per degree C?
That is exactly the question that actually relates back to the resolution. Typically, for example, let's say that you are doing the screen for the first time, and you are just trying to understand what is the TM of your protein of interest after it binds to the ligand. So you ascertain that value. To do that, I would say just start off with probably 10 or 15 acquisitions per degree. Then you will have the full range of your melting curve right there in front of you. Once you figure out, let's say that your TM or your delta TM is probably like 5 degrees, and you picked up your without and with the ligand, probably somewhere around 60 and 65, then you really don't have to start off from, let's say, 30 or 40, and you really don't have to go all the way till 90. So you basically narrow down the melt to around 50 and 80. So, and then you increase the number of data points in that range. So that's how you actually build the resolution, and you can make it up all the way up to 25 acquisitions per degree. And that would be more than enough to actually pick up a resolution as close as a difference of delta TM of around 0.2 degrees. And delta TM of 0.2 degrees, I think it is a high enough resolution to differentiate, especially in the melt curves.
All right. So someone asked, what if your sample is not a set of purified single protein, but rather different mixes? Can you still perform the assays?
It is a very interesting question. I would say with the kind of software that we will be coming probably in the later half of the year, which has got many features in it, you can do data analysis of multiple peaks. Imagine a situation where you have a 100 kilodalton protein, right? And then, so theoretically you wanted to have a 100 kilodalton protein, you purified it, but not necessarily every molecule that is you have isolated out of your purification column is 100 kilodaltons. Some of them are 80, some of them are around 60, and some of them are around 100. So, but all of these would be melting at a different temperature because the TMs or the stability that they attain after interacting with the ligand would be varying. So you will have multiple melting peaks during a situation where you are not dealing with a really highly purified protein. For that thing, you can actually ask the software to define the pre and post melt for a particular peak, and then pick up only that transition event, and then define the TM for that curve. And that can be done in the software that we'll be launching. Probably, I mean, the second version that will be coming out probably like at the end of the sixth month of the year. Yeah.
All right, great. So someone else wants to know, what percentage of proteins have you attempted to perform the thermal shift assays on that are not amenable to this technology?
Our experience with the thermal shift is relatively limited. I would not say that we have done a lot of variations in the proteins and types of proteins that we have studied. But definitely we are actually looking into that direction where we can actually come up with a situation where we can give you an example set for each kind and variations in terms of the proteins.
All right, interesting question here. It says, what are the biggest causes of false positive hits in protein thermal shift assays?
It could be it could be the variables are many here, in the sense like you cannot actually say that this would be the reason for the false positive hits for the thermal shift assays. I would be concentrating more on doing some kind of let me put it this way. A biological truth can be a truth only if more than one analytical events have been confirmed independently. What I mean by that is like you need to have another set of experimentation A and another set of experimentation B, and both the experimentations should be actually confirming the same thing. And then only it becomes a biological truth, and it could be a biological event. And in the situation why you would be having false positives, it could be many reasons. Probably super orange is not probably the concentration of the super orange that has been used is not of the right concentration. Probably we have missed one concentration in the matrix, or the interaction with the ligand, and probably ligand itself is trying to interact with the super orange instead of the ligand interacting with the protein. So it's more than one event that would be contributing for the false positives.
All right, great. We're on our last two questions here. So someone would like to know, can biomagnetic beads or particles bound to the proteins be used in thermal shift assays?
It's a very, very interesting approach. But it actually crossed my thought process when I was looking at the magnetic class particles in terms of interactions. But we haven't tested it. But given a chance, I would definitely want to test those things. But we don't have a particular experience that I can actually comment on that.
All right, and our last question, I think, is a little bit similar to something you talked about before, but someone would like to know, can you slow the ramp rate to 1 degree per minute?
Absolutely. You can go down less than a degree per minute. It is not a limitation on the LightCycler instrumentation at all. The way that we built the LightCycler instrumentation is basically the block actually communicates to the detector. Okay, you can actually take a picture right now. So that message actually comes from the block, and so you can say that I want only 5 acquisitions, and you can actually it ramps at one particular temperature range. And if you want to say you want to pick up something like 25 acquisitions, then the ramping rate would be even slower. So that's how you actually control the ramp rate on that. And absolutely, you can do that on a 1536, and a 480, and a 96 instruments. Yeah.
Great. That's excellent. Well, thanks, Rama. And with that, we've come to the end of our webinar. So I just wanted to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you've missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues. Again, I'd like to thank Rama for his informative presentation, and I'd like to thank you, the audience, for your attention and also some really thoughtful questions. And a very special thank you. Yeah. And a very special thanks to Roche for sponsoring this webinar. So hopefully we'll see all of you again at another Gen webinar in the near future. Goodbye for now.